Let's take a look at the diversity of bacteria. So we're going to be looking at two kingdoms inside of two domains. Originally, we based prokaryotic diversity on like the characteristics that we saw, how they moved, and other visible characteristics. But in the 1970s, we were able to start actually comparing genomes thanks to DNA sequencing, and we were able to take what was once one kingdom called Monera and split it into these two domains. So we have domain Archaea, which are, this is a clade, right? So here would be the common ancestor, and then all the descendants. And domain Archaea is actually sister to the eukaryotes. And then the most diverse or most basal clade over here would be domain Bacteria. And this is the normal kind of bacteria that you would be familiar with. Domain Archaea tends to be bacteria that live in really extreme environments. So let's start with domain Bacteria. This is the domain that's majorly like familiar to most people. And you can see here that this is the first branch point off here. So these guys actually branched off further back than domain Archaea did. Most of these groupings are actually gram-negative. So we've got the Proteobacteria that create this kind of clade up here. The chlamydias have a clade over here. The Spirochetes have another clade over here. And then the cyanobacteria over here, and these are the photosynthetic ones. And notice these all branch off the tree in different kind of places. This is something called an unrooted tree, so we don't have a taxonomic outgroup. But you can see that organisms that branch off closely together like this are going to be more similar to each other. And then we have two kind of groupings of gram-positive bacteria. So gram-positive bacteria is not a true clade, right? Because we have some descendants over here, and then we have some descendants over here. If we wanted to find all of the most recent common ancestors, we'd have to trace this back up here to the common ancestor. But we do have other groupings that kind of come off of this ancestor as well. So the first group, the Proteobacteria, has a bunch of very diverse groups inside of that. Just to touch on a few of these. These are all gram-negative. The alpha group is most closely associated with legumes. So these are going to be like Rhizobium, which is going to live in the roots of legumes like beans and fix nitrogen. We believe that mitochondria were developed through endosymbiosis from some kind of alpha Proteobacteria. The beta Proteobacteria are very nutritionally diverse. Nitrosomonas is used for soil bacteria. It recycles nitrogen. Within this group, we will find Neisseria gonorrhoeae. So that's the bacteria that causes gonorrhea. The gamma group are mostly bacteria that metabolize sulfur. And there are several pathogens in this gamma group as well. We've got Legionella, which causes Legionnaire's disease, Salmonella, which you can often find on, causes food poisoning from chicken, Vibrio, which causes cholera, and then E. coli, which can be your normal gut E. coli, or it can be that pathogenic strain that Chipotle had for a little bit there. We have the Delta subgroup. These are called myxospores. They create these kind of fruiting bodies that look a little bit like a fungus. So sometimes these are called myxobacteria. And then we have the epsilon subgroup, which is typically very pathogenic to humans. There are some bacteria in here that cause blood poisoning. And then this one, Heliobacter pylori, is associated with most stomach ulcers and lives at a really low pH, and so it's able to live inside the human stomach. Our next group we have are the chlamydias. Remember, these guys are over here. And yes, it's that chlamydia. So this is the bacteria that causes the STD chlamydia, which is a common cause for blindness. This is a spirochete. So it's got this spiral shape. So it's a spirilla type of cell. All right, so these spirochetes are over here on our unrooted tree. These are typically helical. They're mostly free-living, but they can be pathogenic. So the bacteria that causes syphilis is a spirochete, as well as the bacteria that causes Lyme disease. Cyanobacteria are photosynthetic, so that's going to be down here. This is like the one we saw already called Anabina, so this is oscillatoria. Again, it forms these kind of large colonies, so each little division here is a cell. So this is going to be a long colony of many, many cells put together, and these are going to cooperate together with some cells fixing nitrogen and other cells doing photosynthesis. We think that the cyanobacteria eventually became plastids through endosymbiosis, so this would be like chloroplasts. And then our last major groupings, again, not a true clade because we've got a group over here and another group over here, are the gram-positive bacteria. Remember, these are the ones with the really thick peptidoglycan cell wall. The gram-positive bacteria rival the Proteobacteria for kind of large diversity. We have things like Actinomycetes, which we used to think was a fungus, but it actually causes tuberculosis and leprosy. Streptomyces, which are typically cultured as a source of antibiotics, so we can actually use some bacteria to develop antibiotics like streptomycin. Bacillus anthracus, which is the bacteria that causes anthrax, belongs to this group, as well as the Clostridium botulism, which causes botulism. Staphylococcus and Streptococcus, which cause things like MRSA, which is methicillin-resistant Staphylococcus aureus, or strep throat from Streptococcus. And then mycoplasms are the smallest known cells, and they are about 5 times the size of a ribosome. They're so small, they don't even have a cell wall, and their genomes typically have about 517 nucleic acids. Not even genes, just nucleic acids. Archaea, that's our next domain. So you can see Archaea over here is sister to Eukarya, which would be the protists, the fungus, the animals, and plants. And these are called extremophile. So phile means loving. These are found in extreme habitats. So halophile meaning salt-loving, thermophile meaning heat-loving, methanogens, so that's what we've got here is a methane swamp. Methanogens are going to live in kind of oxygen-deficient environments where these would be obligate anaerobes that are poisoned by the presence of oxygen. And then they produce methane, and that methane bubbles up as a gas out of these swamps. You don't need to memorize this table, but I think it shows some of the similarities between Archaea and Eukarya. So what we mean by these two being sister is genetically we think that the bacteria in Archaea are more similar to the kingdom of Eukarya than they are to the domain of bacteria. And there's a lot of similarities here. They both have many polymerases. They both start proteins with methionine. They both have introns. Neither are receptive to antibiotics. They both have histones associated with DNA. Obviously, there are some characteristics that are more similar with bacteria, like they don't have a nuclear envelope, they don't have membrane-bound organelles, things that are going to classify them as bacteria and prokaryotic. But it's interesting to see that there are so many similarities between Archaea and Eukarya, and this wasn't something that we discovered until we started looking at genetics in the 1970s. Before that, we thought Archaea was more similarly related to bacteria.